At the outset, I would like to thank the organizers of this conference, the respected chairperson and moderator of the session. My name is Raja Chakraverty, and we will be going towards an aspect of pharmaceutics as well as pharmacology, which is very much required and in vogue today. Now, some of this work at the outset, I would like to place a caveat that some of this work has been done at IPG Kolkata, which is the Institute of Postgraduate Medical Education and Research, and some of this work has also been done at another of the institutes, HIP. So we have been fortunate enough to get grants for this program. So at the outset, uh, I invoke the words of Sir Richard Feynman, who has always said that there's plenty of room at the bottom. So this is a very important term in today's context. Drug delivery and drug discovery are a very important and a protracted process. In the normal scenario, the nanoparticle mediated drug delivery system is something that has caught the attention of the pharmaceutical world. Then my talk today will be focusing around the nanoparticle mediated drug delivery system, especially for the cerebrovascular disorders. Next, please. So this I'm going to run you through the basics of drug delivery and the gaps in current understanding and the pharmacological targets, the blood-brain barrier and its implications in physiology and pharmacology. So what makes an effective target? And also, we will cover the aspect of application in neurology and the way forward. Now, in today's time, we have always seen in cancer therapy, in diabetes, in other very complicated diseases, that there is always a focus on drug mediated in different delivery forms. We have found that there are very good drugs which do not have great bioavailability being sent as a nanoparticle or nanoparticulate system across the blood-brain barrier to improve their permeability across the plasma membranes and the blood-brain barrier. So our studies, the first study was conducted at ICMR NICET, the National Institute of Cholera and Enteric Disease, as a part of our effort to understand the blood-brain barrier better. So that was the first inception point of our understanding. Next, please. So this is one of the pivotal slides. We have always understood that nanoparticles are a very important target, but nanotoxicity is also a very important consideration while assessing nanoparticles and particulate systems. Engineering mechanisms have also been added to target nanoparticles for various kinds of delivery systems. They have very good ability and reproducibility, while it can do away with the risk of systemic toxicity. But at the outset, I should warn the august audience that the nanoparticles itself is not without problem. Nanoparticles and particulate systems can also have nanotoxicity and other problems associated with the delivery forms. We have found that there has been a lot of studies on anti-cancer drugs and their toxicity, mainly myeloid suppression or myelosuppression, as well as alopecia, which is induced or concomitant with. So we will have to understand that any therapy is a two-edged sword, is a double-edged sword. In recent parlance, in toxicology, the first term is do no harm. But these nanoparticles have been shown to do harm also. So we also need, at the same time, effective nanotoxicity assessment techniques, which are being done. There are lots of preclinical, in vitro, as well as in vivo studies on the toxicity of nanoparticles also, because at the end of the day, rather, a drug must be safe, and it must be efficacious. So without efficacy and safety, the advantages that are afforded by a better delivery system crossing the blood-brain barrier have practically very less significance. So the current slide being shown to you is a crux of the matter. We have nanoparticles and various players, various stakeholders, and the crosstalks involving, we can give it through liposomes. We can also target nanoparticles as micellaries. We can also use various other types of carbon-based systems that are in vogue today. We have also understood engineered nanoparticles, solid lipid nanoparticles, gold nanoparticles, and hybrids that are very important in today's therapeutic decisions and choices. But this is also a reality that our studies have some advantages, they also have some disadvantages. I will show you the disadvantages in the upcoming slides. Next, please. So when we talk about the brain delivery, we are talking about a very specialized form of targeting, which has certain advantages. When we target a drug to the brain, we will have to make it lipophilic in nature. We will have to cross the endothelial and the capillary junctions, the tubes, that afford a lot of benefits for pathogen elimination from the body, as well as for the purpose of affording the drugs permeability across and having central effect. So we have to understand that drug delivery to the brain is a two-edged sword. And drug delivery, like in any other part of the body, to the brain is fraught with also a lot of therapeutic decisions, and it is also fraught with a risk-benefit analysis. Whenever we talk about drugs targeted to the brain specifically, we think of the amino acids, we think of the hydrophilic drugs that are also made to enter into the blood-brain barrier, because naturally speaking, these endothelial tight junctions don't afford any mechanism for a hydrophilic drug smell. So the obvious advantage of having hydrophilic drugs run over the blood-brain barrier cannot be overemphasized. So lipophilic drugs obviously can cross this blood-brain barrier. In our studies, we have focused on nanoparticles that could cross the blood-brain barrier. Next, please. When we talk about, this is also one more slide of drug delivery to the brain. This was taken from a very seminal paper by Ehrlich and his associate quite some time back. And it has also recognized that blood-brain barrier, and apart from the blood-brain barrier, there are also other processes of the microglia and astroglia that are very important cells in cell-to-cell communication in neurology today. These have varied applications in neurology, neurosciences, as well as in targeting cerebrovascular disorders. I cannot overstate the problem, because the prevalence rates of cerebrovascular disorders in developing countries is a very high one. And the burden of the disease is also very high in our context. So with that being said, neurological disorders and brain targeting is very important in today's time. This unmet need was assessed by our team. We came out with a review in the Journal of Medical Evidence some time back, and thankfully that has been liked and cited by our colleagues around the world. So that is not the only study that is going on. There are other working groups in various countries that have been focusing on the same problem over and over again. The intranasal route has also come into prominence, where even exosomes and other therapeutic modalities of today's time are also being seen as emerging therapeutics. But I shall warn the august audience again to not be fooled by the concept of a bioactive compound being misunderstood as a drug. Please understand, let's all understand this, that a drug must be druggable. There are drug likeness indices that I am sure that the audience is aware of. Those, for example, the Lipinski's rule of five, like molecular weight, etc., hydrophilicity, hydrophobicity, are all very crucial indicators to support the fact that even a bioactive compound would need to fulfill for being called a drug. So a drug is not only a bioactive compound, it is something that can be given along routes. So drug delivery to the brain, well, what makes it different? It, it is by the sheer complexity of the process and the number of crosstalks that happen in the brain. A small glitch in this regard, in this therapeutics, could mean the difference between life and death, and even harshly impairment of the entire person's life, because a single trigger can influence the person's life in a very detrimental way. Next, please. So this was an idea which was propounded by scientists in Sweden and taken up by scientists all over the world. This focused on the nose to brain delivery of drugs. So the nasal route is very much focused, and in preclinical rodent models of disease also, we have also seen that the nose to brain delivery of drugs have been widely used. This is not something new that I am saying today. There are other people before me who have presented this on numerous occasions in conferences and symposia. But the fact still remains that this is a very important target, but one that is not without disadvantages. We can also remember that during the last COVID pandemic to have hit us, we have also found that vaccine candidates being tried to be used along this route for removal of the virus particles, and not just to limit the extent and the severity of the disease. So the nose to brain delivery of drug and this route has been widely explored in therapeutics and amongst technicians belonging to the pharmaceutical sphere. So nose to brain delivery is very important and it's specialized. We have also, in the process of communicating articles in India and in Southeast Asia on the efficacy of this route. And there are large number of clinical trials also that are studying this in a very important way. So the nose to brain delivery is a very important piece of the puzzle that we are unfolding for brain delivery. Next, please. So this is also very important. The advantages of polymeric nanoparticles. Now, different types of nanoparticles have been shown to have efficacy, organic, inorganic, as well as carbon-based. So polymeric nanoparticles have always shown to be having a controlled drug delivery to the target location, enhanced brain delivery uptake by the tissues concerned. And also, they lower the frequency and the dose administration and a better patient compliance record and having better patient compliance. So these obvious advantages go a long way in ensuring that this kind of polymeric nanoparticles gain eminence amongst the formulation scientists and technicians and technologists and scientists of tomorrow. Next, please. These are the nano formulations based drug delivery system. These are the ones that are available on the net and freely one said that. We have worked, our group has had some experience of working with micelles, some with liposomes. Now we are also targeting antibody drug conjugates. We are also studying about the benefits of exosome and liposome combination. We are isolating, characterizing exosomes and targeting it with nanoparticles. Exosome research is also very new and in the nascent stages in the country. We are also wanting to see whether these kind of nanoformulations can be delivered to the brain and preserve and its architecture. But the question remains drug entrapment efficiency. The question remains the kind of loading or the kind of amount of drugs that actually remains in the part which has been desired and the right organ to for better therapeutic efficacy. If there is subclinical uh, amount or exposure of drugs to a particular part of the brain, that could also mean a failure of therapy. So we have our task carved out and one that is imminent upon us at the moment. Next, please. So transport across barriers. Along with the blood-brain barrier, the blood-placental barrier also has a very important role in therapeutics, in pharmaceutics. So this is also a very important part which needs to be solved by formulation scientists in novel drug delivery systems. And conferences can debate and deliberate and brainstorm all it like. So this will be a very important part also. So we also need such approaches and more brainstorming and data and country-specific data to come up with a solution to these problems. Next, please. Now, the nanoparticle across the blood-brain barrier, this has been explained. There are a lot of advantages of exosomes as well as the neuronal tubes, as well as the dendrimers of these formulations. And we have also seen tagging with various inorganic compounds that have also done wonders to some studies, but these are in nascent stages. These are in lab scale. They have not been scaled up till now. So in sometimes this space is very exciting, and we can have more such compounds in the pipeline before 2028. So there are lots of studies. If you go through clinicaltrials.gov or CTRI, which are the mandates of the clinical trial registration for any kind of drug delivery or drug discovery, you will find a lot of studies which are in phase 1, phase 2 clinical trials based on their claims of coming up with an effective solution with systemic effects and central activity in the brain. So these group of compounds, there are L-carnosine mediated drug delivery systems which involve dexamethasone, and these composites have also found a lot of leverage in drug delivery societies and in various engineering societies. So solid lipid nanoparticles also have had a lot of importance here. So this started off with drugs which were poorly lipophilic. It has continued with even doing something greater. This has also involved biologics. The current revolution of biotechnology and biosimilars have not forgotten that brain delivery of biosimilars is also important for tumors in the brain. A neuroblastoma, for example, has a very less shelf life of the person, and a very aggressive form of cancer can be intervened very quickly and with good outcomes. It can improve quality of life, all-cause mortality of patients if targeted well. So we have to go ahead of the curve in this parlance, in this lexicon, and find solutions that are effective and not toxic. I remind you once again that nanotoxicity is a burning problem. We should not only think about the formulation and the entrapment and the delivery constraints that we are overcoming. We should also think of therapeutics. With the marriage between therapeutics and delivery, you get an effective drug. Let us all remember and reinforce that through our study. Next, please. So in a summary, the optimized or the designing of nanodiversity system will be dependent on the targetability, the drug release, the drug properties, the route of administration. Like any other system, this will have dependence on the biocompatibility, the BCS classification of drugs also for poorly permeated systems, for poorly permeation systems also, there needs to be effective approaches in combination to this. So this is an evolving space with future exciting, such as the ones in AI and ML. Here, AI ML can also be used for effectively understanding and problem solving about the new nascent targets, especially for drugs in phase 1 or phase 2 clinical trials that are there, and just to see the tolerability and the bioavailability of this drug. So with that, I thank you all for your attention, and I would remain open for any questions. Thank you so much.